Okay, so yeah, I'm Johanna. I'm a postdoc with Anne and Etienne, and today I'll be sharing some of the things I've been thinking about with respect to evolution of leaf spectra. Okay, so today I'll be talking mostly about the goal of this project and with an overview of the methods that I'll be using. And I'll illustrate this project using some published studies, using morphometric data, some simulated data, and then some very preliminary, very fresh analyses using the spectral data from Cabo. So like Florence was talking about, previous studies have found evidence for phylogenetic signal in spectral data and have found the predictive power of spectra for either phylogenetic diversity, taxonomic diversity, etc. And so I won't go over the details because we've just seen that. But in general, these studies have tended to use either principal components or principal coordinates or individual wavelengths. So, but no analyses so far have used the full spectra. And so this is kind of the framework that I'm thinking about in terms of using all of the data that we have instead of just reducing the dimensions. So with this in mind, my goal is to calculate the phylogenetic signal for full spectra, fit evolutionary models using the full spectral data, and using these evolutionary models to identify the factors driving variation in spectra, doing some hypothesis testing using these models. And one of the reasons this hasn't been done yet is because we didn't really have methods for it. But in the recent years, a new method has come out, developed by Clavel and Moreland, which I'll talk about a little bit more, which enables this, this research. So overall, the goal is to understand the evolution of leaf spectra, including identifying the best model of evolution for the spectra, and then looking at predictors of variation in spectra. So I'm sure lots of you are seeing yourselves up there. This is the integrative study using data collected for a bunch of different projects through Cabo. I'm using the leaf level spectral data, and I want to thank everybody who was involved in collecting that data. So I have about 2,000 spectra from about 100 species across Canada. And I'm going to be looking at different predictor variables, which include environmental, phenotypic, and phylogenetic variables. So for example, looking at woodiness or growth form, or environmental variables from the plant trait database, such as mycorrhizal associations, nitrogen fixation, and soil characteristics, as a few examples. So the framework that I'm using is phylogenetic comparative methods, and this is necessary because species do not represent independent data points because of their shared evolutionary history. And so this figure on the right from the Felsenstein paper that kind of described this problem illustrates almost a worst case scenario where we have two clusters of species that each cluster has a long branch subtending the clade. And so what this means is that where we might expect to see the pattern shown on the left there, where we have a nice relationship between X and Y, if we count all of those data points as independent, what we actually have is the figure on the right, where we have two clusters of data points, so effectively reducing the number of independent data points we have. And so if we ignore phylogeny, what we end up doing is generating type 1 errors or false positives for these relationships, just because we don't have the statistical support that we think we do. So these phylogenetic comparative methods account for this non-independence due to phylogeny, and the method that's implemented in the newly developed methods is phylogenetic generalized least squares. And I won't go into that into too much detail here. So in addition to accounting for the statistical non-independence of points, phylogenetic comparative methods also let us test different models of evolution to identify how these traits have evolved. So for example, like Florence was talking about earlier, one of the more commonly used models is the Brownian motion model, where this is the phenotype evolves as a random walk along with phylogeny. So if we have an example phylogeny seen on the top there with 3 taxa, the phenotype is illustrated below with phenotype on the y-axis, and you can see that the trait randomly varies over time. And the effect of the phylogeny means that more closely related individuals or taxa have more similar trait values due to this random variation over time. But the Brownian motion model does not include any effects of selection, which is something that we expect given that traits are often under selection, especially the functional traits in the leaf spectra. So another model that we want to look at is the Ornstein-Uhlenbeck model, and this model explicitly incorporates selection. So in this case, we have a random walk with constraints or selection towards an optimum, towards a specific phenotype. So the figure on the right shows phenotype space again on the y-axis, and the Brownian motion is illustrated in black, so that's just random variation. And then the green and the orange show two different Ornstein-Uhlenbeck or OU models. The green one has a lower rate or lower selective pressure, and so you can't actually see, but it has a lower alpha value, which is the parameter, whereas the orange one has a higher alpha parameter, more selection towards the optimum, and this reduces the variance in the trait value towards that optimum value. So those are two of the common methods. There's another method or a model known as the early burst model, which isn't illustrated here, but effectively it's just a deceleration in evolutionary rate over time. So it starts out big burst and slows down over time. But on the right here, we can see a figure from the Morales paper where they illustrate this in spectral data. So the left two columns are Brownian motion. The farthest left is a faster rate, so you get more variation. And the farthest right is an Ornstein-Uhlenbeck model where there's selection towards an optimum. So you can see what that looks like both in terms of the phylogeny and in spectral data itself. Another aspect that is important to consider is heterogeneity across the phylogeny. So we may not expect that there to be a single rate or a single optimum for the whole vascular plant clade, which is the scale that we're looking at. And so we want to be able to test if there are multiple optima or multiple rates across the tree. Okay, so those are the models of evolution. So coming back to our spectral data, these include some reasons that this has not been done before, primarily because of the high dimensionality of the data. So we know that spectra has around 2,000 or so individual wavelengths, which means that the number of traits that we have is much greater than the number of taxa that we have. And this causes statistical problems with the typical methods of using the full likelihood approach. So this method developed by Clavel and Moreland uses a penalized likelihood approach, which should escape that problem. We also have autocorrelation between the individual wavelengths, and this method also accounts for autocorrelation because it's developed for morphometric data, which has the spatial element as well. The method also allows us to evaluate non-Brownian motion models, which is something that isn't possible in a lot of other methods. They only include Brownian motion. So here we can test the OU model, which according to Morales et al., they think that the OU model is the best fit model for individual wavelengths. So we suspect that may be the case for the full spectra as well. So given the similarities of spectra to morphometric data, we feel like this approach is well suited for spectral data. So now I'll go over the specifics of the methods that I'll be using. So we have our multivariate trait data, which is the spectral data. We have a phylogeny. Right now today, I'm just showing one example of phylogeny, but I'm hoping to have a distribution so we can look at uncertainty in the phylogeny itself as well. And then we have our predictor variables. So this is like the soil or woodiness, et cetera, that I talked about before. So we take these input and do a generalized least squares multivariate analysis, basically doing a phylogenetic regression on these input. And then we can get out a number of different things. So the first thing is model parameters. So like I mentioned, there could be different strengths of selection towards an optimum. There could be different rates of evolution. And that is the first kind of piece of output that we can get from these models. The second thing we can do is hypothesis testing using MANOVA, using the model fit for the data. And then we can also generate phylogenetic PCAs using the variance-covariance matrix used in the model fit. And I'll talk more about that a little bit later. Another thing you could use these models for is to generate ancestral state reconstructions. So you can reconstruct what the ancestral spectra was at some point on the tree, if that's something of interest. So what do I plan to do? So this is where I'm kind of integrating some published studies using morphometrics, some of my very preliminary analyses, and then some references to simulations as well. So the first step is identifying the best fit evolutionary model for the spectral data. So these are two examples of using morphometric data where they identified the OU model as the best fit model. And in the example on the right, they report the alpha or strength of selection, which is actually a fairly small number. So there's pretty weak selection in their morphometric data set. And so yeah, we hypothesize that the OU model will be best for the spectral data as well. The next thing that you can do is assess the impact of these predictors on the spectra. So in this case here, they looked at the evolution of cranium shape. And one of the predictors that they looked at was habitat. So they identified that habitat was a significant predictor of cranium shape. So in 2 different habitats, there are 2 different shapes of cranium in general. And this could be due to selection for these different shapes. And this is conducted using the penalized likelihood MANOVA on those models. So here's where I did some very preliminary analyses looking at spectra, and I was using the pattern that Florence was just talking about, the conifer versus angiosperm pattern, where there are differences between those 2 groups. And so this is what's illustrated on the right. The green is conifers and the purple is angiosperms. And so using this phylogenetic approach, I fit the model and then did a MANOVA for assessing spectral differences between angiosperms and conifers. And you'll notice that that was not significant. And this is because this is where I need to be careful with my words. This is a phylogenetic comparative method. And so the method accounts for phylogeny in the model fit. And so basically what has happened here is the variation between conifers and angiosperms is accounted for in the model. So once that's accounted for, there's no more remaining signal of angiosperm versus conifer. So it seems a little bit counterintuitive, but what it actually means is it's doing what we think it's doing. It is taking that variation due to phylogeny and then fitting a model that does not include that phylogeny, if that makes sense. I don't think I explained that very well. But so what we see is, yes, there was a very strong impact of phylogeny on the difference between conifers and angiosperms. And now we have a model where that is not driving the patterns that we see. So now we can use this model to test other relationships. So if, for example, with woodiness, if we saw a pattern of woody versus non-woody, that wouldn't be due to the phylogeny, that would be due to the difference between woody and non-woody plants. Hopefully that makes sense. Okay, so that was kind of the first stage of the project. And once we have those, the models for the evolution and looked at some predictors of the variation, we want to look at identifying the primary axes of variation, and then looking at drivers of evolution of those individual axes. So the first thing is to use the model that we fit in the variance covariance matrix to identify primary axes of variation using the phylogenetic principal components. So here we have the same data. We have angiosperms in purple and conifers in the green triangles, and we can identify the primary axis of variation. And then once we have these axes, we can model the evolution of these axes. And one of the reasons we wanted to reduce the dimensions basically is that the full methods cannot test between more complex models. We can only test the simplest versions of these models. So for example, a single rate or a single optimum. But if we reduce the dimensionality to these principal components, we can test some of these more complex models, such as single versus multiple rates or single versus multiple optima. And this is what was done in another morphometric study here, worked with the first 3 principal components. They identified different evolutionary models and linked that to the pollination syndromes, which was selecting for different shapes, primarily on principal component 1. So we can link these models to the underlying traits underlying these principal components. So for example, if we were to use the example of the conifer versus angiosperm differences, we might expect that this would be driven due to the structure of the leaves, the needles versus the broadleaves. And so would we see differences between the conifers and angiosperms on the principal component comprised of wavelengths related to structural elements? And this is where it kind of ties into what Sean's doing as well. And we look, is there selection maybe for different spectral optima in those wavelengths? This can tie back to the traits from the principal components. And then the next and kind of last step is to assess the impact. And so, for example, in this study here, which was on a different group of plants looking at flower shape as well. So what they found was 3 different optima for the color here, which were different from the optima overall for the tree. And they were able to say that because these optima went to the right on the principal component, that this was actually associated with an increase in corolla length compared to the others. So we're able to look at the impact in the actual trait value on these principal components. So that's all I have kind of for the analysis so far, but I'm also doing some simulations, partly because our data set is so much larger than the morphometric data sets, just to make sure that it works as expected. So the first step of this is to simulate the evolution of a single parameter. I'm going to be using the structure parameter under different models of evolution. So on the left, you can see that Brownian motion model, and on the right is OU model, where I've simulated for a 10 taxon tree this structure parameter. I input the structure parameter into the prospect rate of transfer model and generate these evolved spectra. So here we can see the spectra in brown have evolved under Brownian motion, and the spectra in green have evolved under Ornstein-Uhlenbeck, where you can see it has a much smaller range of variation due to selection towards this optimum. Then the next step will be to use these simulated spectra and model them using the approach that I described earlier. I know what the best model should be, and I know what the parameters should be, so I should be able to see, am I getting what I expect to? This is just one iteration of a very preliminary version, and so far it doesn't look like we quite get what I expect, but it's a work in progress. So if you have experience simulating spectra, maybe talk to me afterwards. So yeah, very much a work in progress. So what do we want? What do we expect to get out of this? We expect to identify the evolutionary model for the full spectra, identify primary axes of spectral variation and the underlying traits, and then be able to test hypotheses for drivers of the spectral variation for both the full spectra and for individual axes. And with that, thank you. And I guess maybe if there's questions, I have a little bit of time.